A simple 5-carbon molecule gives the lemon its signature citrus scent.
Just 5 carbons, yeah.
Right. But if you take a few more of those exact same 5-carbon molecules and, stack them together, you construct the cholesterol that can clog a human artery.
Which is pretty wild to think about.
It really is. Welcome to the Deep Dive. Today we are exploring Paul Amdoak's definitive textbook, Medicinal Natural Products, to uncover the hidden biochemical architecture of life-saving drugs.
It is a fantastic resource.
Yeah, and if you are a medical student or just a professional in the healthcare field, this deep dive is completely tailored for you.
Yeah.
We are going to deconstruct how nature's pharmacy actually works.
Right, moving past just, memorizing drug names.
Exactly. We want to understand the precise molecular machinery that builds these compounds from scratch.
And to really understand that machinery, we have to look at how an organism manages its resources. I mean, every living thing, whether it is a plant, a fungus, or a human, relies on primary metabolism.
Keeping the lights on, basically.
Right, exactly. These are the universal, non-negotiable pathways. Things like glycolysis and the Krebs cycle. They break down food, generate ATP, and synthesize the components needed for, well, just basic survival.
But the drugs we actually use in the clinic, those almost entirely originate from what we call secondary metabolism.
Yes.
And secondary metabolism is it is essentially the organism's specialized defense budget, correct? Like it is not about basic survival, it is about evolutionary advantage.
That is the perfect way to frame it. I mean, organisms can't just run away from predators or infections, so they wage chemical warfare.
They stand and fight.
Right. They produce secondary metabolites to act as toxins, repellents, or attractants. And to build these specialized weapons, the organism taps into the intermediates generated by its primary metabolism.
Because nature is incredibly efficient.
Extremely efficient. It doesn't invent entirely new chemical elements for these defense systems. Instead, it relies on just 4 primary building blocks.
And those form the 4 distinct biosynthetic pathways. So for you listening, we have acetyl-CoA feeding the acetate pathway. We have shikimic acid, which comes from carbohydrate metabolism.
Right.
Then there is mevalonic acid and deoxycallulose 5-phosphate for the terpenoids and steroids. And finally, standard amino acids, which provide the foundation for alkaloids and peptides.
And if you look closely at the structure of any complex medication derived from nature, you can reverse engineer it right back to those 4 foundational blocks.
That is fascinating.
It is. The most fundamental of these is the acetate pathway. It uses a remarkably simple 2-carbon unit derived from acetyl-CoA to build an astonishing variety of structures.
And the underlying mechanism here relies on Claisen condensations.
Yes, exactly.
So for those trying to visualize this, a Claisen condensation is basically a chemical stitching process. You are taking a 2-carbon molecule and coupling it with another, creating a 4-carbon chain.
Then a 6-carbon chain and so on.
Right. But crucially, this process leaves behind highly reactive oxygen groups along the chain, forming what is called a polybeta keto chain.
And the fate of those reactive oxygen groups, that determines whether that organism produces a normal dietary fat or a potent drug.
Wait, really? Just the oxygen groups?
Yeah. In standard fatty acid biosynthesis, specific enzymes systematically reduce those oxygen groups. They strip them away at each step,
leaving behind a flexible, straight hydrocarbon chain.
Exactly. Those fatty acids eventually become part of the cell membrane, or, they are converted into arachidonic acid in humans.
Which brings us directly to clinical pharmacology. Because arachidonic acid is the immediate precursor to prostaglandins, thromboxanes, and leukotrienes.
Right, the inflammatory mediator.
Yeah. So when you prescribe an NSAID, a nonsteroidal anti-inflammatory drug, you are actively blocking the cyclooxygenase enzyme from converting that flexible acetate-derived chain into the mediators of pain and inflammation.
Exactly. But the clinical applications shift dramatically when the organism decides not to reduce those oxygen groups.
And when it leaves them alone.
Right. In secondary metabolism, polyketide synthase enzymes take that highly reactive polybeta keto chain and stabilize it without reducing it.
Oh, wow. So because those oxygen groups are still intact, the chain can fold back on itself.
Yes. The functional groups react with one another. They stitch the linear chain into a series of rigid, fused aromatic rings.
So the difference between a flexible fat and a rigid multi-ring drug is just whether the biological factor decided to hit the reduce button during assembly.
Pretty much. It highlights the extreme modularity of these biological factories. When that chain folds into rings, you get aromatic polyketides.
Okay.
This is how plants produce anthraquinones, the active compounds found in senna and cascara.
The classic stimulant laxatives.
Exactly. The folding pattern determines the pharmacological action, and we see an even more complex folding pattern in the tetracycline antibiotics.
Oh, from the Streptomyces bacteria.
Right. Tetracyclines contain a dense four-ring skeleton built almost entirely from simple malonate units, but initiated by a unique acetyl-CoA starter molecule.
So, if the folding pattern is dictated by enzymes, it stands to reason that we could intercept that process, right? Like, if we know exactly which enzyme folds the ring or adds a specific atom, we could hypothetically modify the organism's genetic code to build a customized drug.
Oh, that is actively done in pharmaceutical manufacturing today.
Really?
Yeah. Take Streptomyces aureofaciens, which naturally produces chlortetracycline. By genetically knocking out the specific halogenase enzyme responsible for adding the chlorine atom during the final stages of the assembly line, the bacteria is forced to produce standard tetracycline instead.
That is incredible. We are physically altering the assembly line to optimize the clinical profile of the antibiotic.
Exactly. We are hacking the factory.
So the acetate pathway is brilliant for building these fused ring systems from long carbon chains, but nature often requires a totally different structural approach.
Right, like with wood.
Yeah, wood needs to be incredibly rigid, and that requires massive amounts of stable aromatic rings. And the acetate pathway is, well, it is actually quite inefficient for churning out standalone aromatic rings on a massive scale.
It is. And that structural demand is what drives the shikimate pathway. It is nature's alternative route to aromatics.
So how does that differ?
Instead of stitching two carbon units together, the shikimate pathway pulls directly from carbohydrate metabolism. It grabs erythrose 4-phosphate, which is a 4-carbon sugar.
And phosphoenolpyruvate, the 3-carbon molecule.
Right. By combining them, the plant synthesizes the aromatic amino acids, phenylamine and tyrosine.
And those amino acids are obviously crucial for building proteins, but they also serve as a vital architectural template, right? The C6C3 unit.
Yes. You have a rigid 6-carbon aromatic ring attached to a 3-carbon tail.
Right.
By removing the amine group from phenylalanine and tyrosine, the plant creates cinnamic acids. And these C6C3 units are incredibly versatile.
They really are. They can be cyclized to form coumarins, which are the structural basis for anticoagulants like decumarol.
Exactly. But the true pharmaceutical heavyweights in this category are the lignins. They are formed when two of these C6C3 units undergo oxidative coupling.
So they link together at their tails.
Yes.
The prime medicinal example of a lignin is podophyllitoxin, extracted from the podophyllum plant. And this molecule has a fascinating, if, brutal mechanism of action.
Brutal is a good word for it.
It physically wedges itself into the hydrophobic pocket of the protein tubulin.
Right, and tubulin is the primary building block of the mitotic spindle.
Which you need for cell division.
Exactly. During cellular division, microtubules have to rapidly assemble and disassemble to pull the chromosomes apart. When podophyllitoxin binds to tubulin, it prevents that polymerization.
The cell is just frozen in metaphase.
Completely frozen. The mitotic spindle simply cannot form, and the cell undergoes apoptosis.
Halting cell division is obviously the foundational goal of oncology, but throwing a systemic wrench into the mitotic machinery of every cell in a human patient is devastatingly toxic.
It is. Podophyllitoxin proved far too dangerous for systemic chemotherapy. Gastrointestinal toxicity and neurological damage were just severe.
So what did we do?
Well, organic chemists used that natural structure as a blueprint. Through semi-synthesis, they added a bulky glucose derivative to the molecule, creating adiposide.
Oh, adiposide.
Yeah, and this structural modification changed the drug's mechanism entirely. Adiposide no longer targets tubulin.
Wait, it doesn't?
No, it targets topoisomerase II, which is an enzyme that manages DNA tangling. It traps the enzyme after it has cut the DNA.
Causing lethal double-strand breaks and rapidly dividing cancer cells.
Exactly, but with a significantly safer toxicity profile for the patient.
It is a perfect example of human ingenuity upgrading nature's baseline defense mechanisms. We take the poison, alter the three-dimensional geometry slightly, and create a targeted therapy.
Which provides a great transition into our third major pathway, the terpenoids and steroids.
Right, because here we move away from flat aromatic rings and enter the world of complex three-dimensional stereochemistry.
And this domain is governed by the isoprene rule. The mevalonate and deoxyxylose phosphate pathways synthesize a simple 5-carbon building block called an isoprene unit.
And the sheer biochemical economy here is staggering. Plants and microbes simply take these 5-carbon units and link them head to tail, continually adding to the chain to create vastly different molecules.
And we categorize them purely by how many of those 5-carbon blocks are linked.
Right. 10 carbons give us monoterpenes. 15 carbons create sesquiterpenes, which include massive therapeutic agents like artemisinin.
The frontline defense against malaria.
Exactly. And then adding another 5 gives us the 20-carbon diterpenes.
Which include paclitaxel, commonly known as Taxol, isolated from the bark of the Pacific yew tree.
And Taxol actually does the exact opposite of podophyllitoxin, right?
It does. Instead of preventing tubulin from assembling, Taxol hyperstabilizes the microtubules. It locks them in place so they cannot disassemble.
Which also freezes the cell in mitosis and triggers cell death.
Exactly. Just approaching the problem from the other direction.
Now, the versatility of that 5-carbon block becomes truly apparent when we reach 30 carbons, the triterpenes, because this is where the linear chains undergo a massive concerted cyclization event to form the steroid skeleton.
The steroid skeleton is a marvel of spatial architecture. In mammals, that 30-carbon precursor becomes cholesterol, which is modified into the steroid hormones.
Right.
The foundational structure is often the C21 pregnine skeleton. In this highly rigid 3D scaffold, the tiniest enzymatic tweak, adding a single hydroxyl group or changing the angle of a double bond, completely alters the molecule's affinity for specific receptors in the human body.
And we see that stark contrast in the adrenocortical hormones like hydrocortisone. A glucocorticoid has a specific oxygenation pattern that makes it bind tightly to glucocorticoid receptors, powerfully suppressing the immune system and altering carbohydrate metabolism.
Right.
But if an enzyme just oxidizes one specific methyl group into an aldehyde, you get aldosterone.
And aldosterone is a mineralocorticoid. That single oxygen atom shifts the receptor binding profile entirely.
Just one atom.
Just one. Instead of managing inflammation, aldosterone binds to receptors in the distal tubules of the kidneys, commanding the body to retain sodium and excrete potassium,
directly controlling blood pressure and electrolyte balance.
The biological effect is night and day, all based on a microscopic change in the functional groups decorating that steroid scaffold.
And that spatial precision is even more critical in the cardiac glycosides, like digoxin sourced from the digitalis plant. The steroid core in these molecules doesn't look like a typical human hormone.
No, it doesn't. The stereochemistry of cardiac glycosides is highly unique. The 4 rings of the steroid nucleus, labeled A through D, are fused in a very specific pattern.
How so?
The A and B rings are cis-fused, and the C and D rings are also cis-fused. This forces the entire steroid molecule into a distinct, rigid U-shape.
And that U-shape is not an accident. It is the exact physical key required to plug the extracellular channel of the sodium-potassium ATPase pump in myocardial cells.
Furthermore, cardiac glycosides have a critical hydroxyl group at the 14-beta position, pointing in a specific 3-dimensional orientation.
Which forms a vital hydrogen bond with the ATPase pump.
Exactly. By jamming this pump, sodium builds up inside the heart cell. The body tries to compensate by treating that sodium for calcium.
And that sudden influx of intracellular calcium is what vastly increases the force of the heart muscle's contraction.
Right, providing the cardiotonic effect used to treat congestive heart failure.
It is amazing how physical geometry dictates clinical outcomes. Up to this point, we've mostly been discussing molecules built entirely of carbon, hydrogen, and oxygen.
Right.
But when nature decides to inject a nitrogen atom into these structures, the chemical behavior of the molecule fundamentally changes. Which brings us to the 4th major biosynthetic category, the alkaloids.
Nitrogen is an absolute game changer. It typically makes the molecule basic or alkaline, which is where the term alkaloid originates.
Okay, that makes sense.
Because they are basic, alkaloids often exist as positively charged ions at physiological pH. This charge allows them to easily cross the blood-brain barrier and bind with incredibly high affinity to the negatively charged pockets of neuroreceptors.
And nature synthesizes these alkaloids by just hijacking standard amino acids, right?
Pretty much, yeah.
So if we trace ornithine, we find the precursor to the tropane alkaloids. This family includes hyoscyamine and hyoscine, famously extracted from Atropa belladonna, the deadly nightshade.
Right, and these compounds are competitive antagonists of the muscarinic acetylcholine receptors.
They blockade the parasympathetic nervous system,
leading to pupil dilation, tachycardia, and, potentially fatal delirium.
The line between a therapeutic agent and a lethal poison here is exceptionally thin. It often comes down purely to pharmacodynamics and the route of administration, rather than the molecule itself being inherently good or bad.
Oh, absolutely. The tyrosine derivatives offer a brilliant case study on that precise line.
The ipecacuanha plant.
Yes. It produces the alkaloids amethine and cephaline. And at a cellular level, these molecules are just catastrophic. They bind to the 40S ribosomal subunit in eukaryotic cells, completely halting the translocation phase of protein synthesis.
And if a cell cannot synthesize proteins, it dies. So using amethine systemically to fight an infection or a tumor would cause massive indiscriminate cellular death across the patient's entire body.
Systemic administration is highly toxic. However, for decades, ipecac syrup was a staple in emergency medicine for treating oral poisoning.
Because it induces vomiting.
Yes. When swallowed, emmetine and cephaline act as severe, localized irritants to the gastric mucosa, while also stimulating the chemoreceptor trigger zone in the medulla.
So the intense local toxicity forces the stomach to violently empty its contents.
It is a perfect example of weaponizing a plant's lethal defense mechanism to expel other toxins.
It is entirely about clinical context. And the tryptophan derivatives are equally impactful. They yield the indole alkaloids, which include reserpine from the Rauwolfia plant.
Which completely changed cardiovascular medicine as the first major antihypertensive.
Working by depleting monoamine neurotransmitters from nerve terminals. We also see ibogaine in this class, right?
Yes, a potent psychoactive alkaloid currently under intense research for its ability to interrupt opioid addiction pathways in the brain.
And in every one of these alkaloid classes, the nitrogen atom, donated by the original amino acid, is the structural linchpin that dictates how the drug interacts with the human nervous system.
Exactly.
Which sets up our final and perhaps most complex structural category. We just explored how nature modifies single amino acids into alkaloids. But what happens when microbes start stitching entire chains of amino acids together into massive, customized macromolecules?
Then we enter the realm of peptides, proteins, and the beta-lactam antibiotics.
So anyone in the medical field is intimately familiar with ribosomal peptide biosynthesis. The ribosome reads messenger RNA and strings together the 20 standard proteinogenic amino acids.
Right. This is how the human body produces insulin, growth hormone, and calcitonin. It is highly regulated, but it is also strictly constrained by the genetic code.
A ribosome can only build with the 20 basic amino acids it is programmed to recognize. It cannot spontaneously decide to throw in a bizarre, modified chemical structure.
No, it can't. But microbes bypass this limitation entirely through secondary metabolism.
How did they do that?
They evolved a separate, incredibly sophisticated system called non-ribosomal peptide biosynthesis. This relies on an enzyme complex known as a non-ribosomal peptide synthase, or NRPS.
I want to make sure the mechanics of this are clear for you listening. An NRPS is not reading an mRNA template, is it?
Not at all.
It is a physical modular assembly line. Think of it like a massive robotic arm in a car factory. Each module in the NRPS is responsible for grabbing one specific amino acid, modifying it, and passing it down the line.
And because it operates independently of RNA, the NRPS can incorporate hundreds of unusual non-protein amino acids, including D-amino acids, which standard ribosomes completely ignore.
It can cyclize the peptide chains into rings, and it can perform radical structural modifications.
The most famous example of this is the biosynthesis of penicillins and cephalosporins by fungi and soil bacteria.
The defining feature of those antibiotics is the beta-lactam ring. And that ring is something a standard ribosome could never synthesize.
Exactly. The NRPS takes 3 precursor amino acids, aminodipic acid, cysteine, and valine, and fuses them together. Then specialized enzymes aggressively oxidize the chain, forcing it to close into a 4-membered beta-lactam ring.
And from a chemistry perspective, a 4-membered ring is highly strained. The bonds are forced into unnatural angles, meaning the ring is desperate to pop open. It is essentially a loaded spring.
And that structural strain is exactly what makes it a deadly warhead against bacteria.
How does it detonate?
Well, when a bacterium attempts to build its cell wall, an enzyme called a transpeptidase reaches out to cross-link the peptidoglycan layers. The transpeptidase mistakenly attacks the beta-lactam ring.
And because the ring is so strained,
it immediately springs open and forms a permanent covalent bond with the active site of the transpeptidase.
the bacterial enzyme is irreversibly handcuffed. The cell wall weakens, and the bacteria undergoes osmotic lysis and just explodes.
You really cannot understand pharmacology without understanding that spatial geometry. Tracing these pathways, whether it is the folding of an acetate chain, the rigid aromatics of the shikimate pathway, the 3D scaffolds of terpenoids, or the massive NRPS assembly lines, it moves our understanding of medicine from simple memorization to mechanical comprehension.
You start to see why a drug binds to a specific receptor, why a slight modification changes its toxicity, and how evolution engineered these molecules in the first place.
And that mechanical comprehension opens up a fascinating future for pharmacology.
It really does.
I mean, if these NRPS and polyketide syntheses are just modular assembly lines governed by discrete sets of genetic instructions, the implications for synthetic biology are massive.
Oh, for sure.
The field is already moving in that direction. We are currently sequencing the genomes of uncharacterized deep-sea bacteria and using AI systems like AlphaFold to predict the physical structures of their undiscovered NRPS modules.
That is mind-blowing.
In the near future, we won't just be discovering new drugs in nature, we will be using genetic engineering to mix and match these biological modules. We could program a yeast cell with a custom-built NRPS to physically print a novel, highly complex antibiotic that evolution never even thought to invent.
We were transitioning from harvesting nature's pharmacy to actively reprogramming it. To solidify everything we've covered today, I have a quick mental exercise for you to try before you head into your next clinical rotation. Trace the biosynthetic origins of two very common drugs, a corticosteroid, like hydrocortisone, and a tetracycline antibiotic. Which of the four fundamental building blocks does each drug use, and how do their primary metabolic precursors dictate the final shape of the molecule? Understanding that core difference is the key to mastering the molecular landscape of medicine. Keep digging into the science behind the prescriptions, and we will see you on the next deep dive.